Hey everyone, it's Miss Bell again. So today we're going to be talking about DNA, RNA, and protein synthesis, and then later on for this unit we'll be talking about mutations in DNA, and then also some technologies that we are using today in DNA, with DNA. So anyway, we all started studying DNA back in the 40s. Watson and Crick are known for their studying DNA. So anyway, James Watson, he was an American, and Francis Crick was from England, and they were studying DNA together at Cambridge University in England. And then in the 50s, in 1953, they successfully described the structure of DNA, and they proved that it was the basis for heredity among every living thing. Bacteria, plant, animals, everything. Because of their research, they won the Nobel Prize in 1962. So everything we know about DNA and even RNA started with those guys. So anyway, another important person, anytime we cover people, you need to make sure you know them and what they did. Another important person is a female. We don't hear a lot about females in science because many females were not scientists back in the day when all this stuff was discovered. But anyway, so this female is Rosalind Franklin. So Rosalind Franklin, she also studied DNA. And she used something called X-ray diffraction. And she is the person who figured out that a chromosome basically is DNA. Okay, so that a chromosome is DNA spiraled together to make the chromosome shape. She also discovered that it had a sugar phosphate backbone. So as a refresher, we've talked about chromosomes and DNA a little bit before in a couple of different units, in the biomolecules unit and also in the cellular reproduction unit. But anyway, just a refresher, chromosomes are DNA. Chromosomes are just DNA that's made into an X shape. Okay, so it's like a slinky, the DNA is. And if you push the slinky together, then you get a chromosome. If you pull the slinky out, then you get chromatin. So chromosomes and chromatin are the two shapes that DNA can take. But it's still going to be a spiraled ladder either way. It's just going to be either condensed or not. Because with a slinky, it's still a spiral ladder, even if you condense it. But anyway, so this is just a reminder that each chromosome is a long DNA, and on that DNA, you have many genes. Okay, so another thing you need to know is that DNA controls the production of proteins in a cell. Now, we talked about DNA containing all your genetic information, and it's kind of like where the control comes, but how it does that is by making proteins. Anytime you make a protein, things happen because of that protein. Now, one gene is one piece of DNA, so it's a tiny segment of DNA, and that's part of a chromosome. So you have 46 chromosomes in your body cells, like we learned in the last unit. Each one of those chromosomes has hundreds or thousands of genes. Okay, each of those genes is responsible for one of your inherited traits, like your eye color, hair color, skin color, etc. So, when DNA directs the protein production in the cell, it then expresses a gene. So, when you make a protein, you express a gene. That is how your traits are seen on the outside. Okay, so think about it. Proteins make up the structure of most things in your body. Okay, so if you make a certain protein, you'll see a certain trait. Like your eyes, for example, your eye color, different eye colors are because of probably different proteins. So because of that, you'll see a certain eye color. So this picture is to show you what I was just mentioning before. So this up here would be like your slinky that is condensed, and this would be like your unwound slinky. Notice it's still a double helix. Okay, so DNA is still a double helix. It just coils into a chromosome shape or chromatin. So DNA stands for deoxyribonucleic acid. And it's named for that because deoxyribose is the sugar in this particular nucleic acid. So, as far as the parts of DNA goes, you have, it's like I said earlier, it's a spiral ladder, also known as a double helix. Okay, so your sides of a ladder, are going to be made up of sugars and phosphates. Now, specifically, it's in DNA, it'll be deoxyribose, which is a 5-carbon sugar. Okay, so C5H10O5, because it's still going to be in a 1:2:1 ratio because it's a sugar. Anyway, so you have a sugar phosphate backbone. So what you'll have is your 5-sided sugar and then a phosphate, and then a 5-sided sugar, and I'm not a good drawer, and then a phosphate. So it'll be sugar, phosphate, sugar, phosphate. So that's the whole sides of DNA. Sugar, phosphate, sugar, phosphate, sugar, phosphate. So that's the sugar phosphate backbone. Backbone, that's what they call it. And so the sugar is always deoxyribose in DNA, and then it's just a phosphate group. Now, the rungs, which are going to be the horizontal portion, are made up of nitrogen base pairs. You see how there's 2 of them? Okay, and then they are going to be bonded together with a hydrogen bond. Okay, now those base pairs in DNA are either going to be A and T or C and G. And I could flip them and put the G first and then the C, or the T first and the A. But in DNA, you have adenine, thymine, cytosine, and guanine. So those make up the rungs of the ladder. So this is a great picture showing the structure of DNA. Okay, so I want to point out one nucleotide. If you recall, a nucleotide is a sugar, a phosphate group, and a nitrogen base. So one nucleotide would be 1 nitrogen base, 1 sugar, and 1 phosphate group. Okay, so here's your phosphate, here's your 5-carbon sugar, and then here is your nitrogen base. Okay, so that's one nucleotide. So if you look, you've got many nucleotides that make up a DNA molecule, and that's why nucleotides are the monomer of DNA. So if you'll notice, you have your hydrogen bonds in the center, and you are bonding your adenine and your thymine to make up one rung of a ladder. And like I said, you have a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and that makes up your rails or your backbone. So here's just another picture of the sugar phosphate backbone over here. They've zoomed in on it, and it's showing you your base pairs, and you'll have a hydrogen bond between there. So again, DNA is made up of nucleotides. If you recall, nucleotides are the monomer of DNA, or nucleic acids. DNA is a molecule that's common to every living thing, no matter what, if it's prokaryotic or eukaryotic. So if it's prokaryotic bacteria, it still has DNA. If it's a eukaryotic cell, it still has DNA. Okay? It is considered the blueprint, so it has all your genetic instructions for building those proteins. Okay? So it contains the genetic instructions for building proteins. When you build a protein, that allows your genes to be expressed, or for you to be able to see your genes on the outside. Now, you do have some variations in your nitrogen-based sequences, and that's going to vary from person to person, and it's going to vary from species to species. Okay, and those differences are what's going to make you and I different from each other. So the only thing making you and I different is those nitrogen bases are just in a different order for you and I. but they will be closer together than, say, if you were to compare my DNA to a turtle. I'll have more similarities with another human than I would with like a turtle. But my DNA would not be the same as another human because we are different. So, in every species, the amount of adenine, A, equals the amount of T, thymine, and the amount of G equals C. That's how they know that these base pairs, that the bases will pair up. All organisms contain chromosomes made up of DNA that have the nucleotides with adenine, thymine, guanine, and cytosine. So even bacteria will have DNA just like yours with sugars and phosphates and those nitrogen bases A, T, C, and G. Okay, but what makes us different from each other and different from other organisms is because the order of nucleotides. Okay, the order or sequence of nucleotides. Okay, so for example, I might be A, C, 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 G. You might be A, G, C, 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 G. Okay, so that might be the difference between us. The order or sequence of those nucleotides are going to be just a little bit different. Now, this is me. This is you. A frog might be A, G, A, C, 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 G. Okay, so they might have more differences. But anyway, we are different because even though we're made of the same things, they're all put in a different order. Okay, so this is how squirrel is different from a rose bush because the order of the nucleotides is different. And so this would be the order of the nucleotides, okay, or your order of nitrogen bases. So this is called the variability of base sequencing. Okay, so variability is like the differences or the amount of variety. One, okay, A stands for one. One human chromosome contains 140 million base pairs. That's one of your chromosomes. Remember, you have 46. Since any four of your possible nucleotides, so that would be A, T, C, or G, can be present at any of those 140 million base pair positions, your total number of possible sequences per chromosome now, and remember you have 46, is 560 million. Okay, so there's a 1 in 560 million chance that you would have one of the same sequences as someone else. Now, imagine this for all 46 chromosomes. Okay, so that's when we get this staggering number here. Okay, we don't have that many people that exist on Earth. Anyway, right now. So, or I guess we do, but you still have the chances of the egg and sperm and remember crossing over and all that. So, anyway, one DNA molecule or a DNA molecule cannot be viewed with a compound light microscope. Remember, we talked about that before. It can only be seen with an electron microscope, and if you recall, it can only be seen during mitosis. Usually prophase or metaphase is the best time, and it can never be seen during interphase. So again, DNA is made of nucleotides. Nucleotides are the monomer of nucleic acids, and DNA is one of the two nucleic acids. So, in a nucleotide of DNA specifically, you're going to find the same things in every nucleotide: a sugar, a phosphate group, and a nitrogen base. But specifically, the sugar will be DNA. In DNA, deoxyribose, and in RNA, it'll be ribose. Okay, make sure you know that. And then you'll have your phosphate group. And so the sugar and phosphate group bond together to make up your sides of your ladder or the rails. Okay, and then your rungs, remember the rungs are the horizontal portion or side-to-side portion, is made up of your nitrogen bases. In DNA, your nitrogen bases are adenine, thymine, A and T, guanine, and cytosine, G and C. Okay, and they are bonded together by hydrogen bonds. So one rung is made up of two nitrogen bases bonded by a hydrogen bond. And then you'll have your sugar and your phosphate. And then your sugar, actually the sugar should be attached to the nitrogen base. Whoops, do it like that. Anyway, y'all don't have to know it that specific. You get the picture. So anyway, here we go. We have our picture of DNA. They've straightened the ladder out so it's easier to see. And so notice you have your sugar phosphate backbone here on the side. Okay, sugar phosphate, sugar phosphate, sugar phosphate. And then I can see here A and T, so this must be DNA. And they are bonded here by a hydrogen bond. Okay, so you got your A and T's bonded up and your G's and C's. So it takes two nucleotides. So remember, you got your phosphate group, your sugar, your nitrogen base. So there's one nucleotide, and then here's another nucleotide. So it takes two nucleotides to make one rung on the ladder. So base pairing. So base pairing rules. Nitrogen bases can be either a purine or a pyrimidine, okay? And if you go back to the picture, you can notice how they're shaped different. And if you flip back, you can see one's kind of bigger than the other. But anyway, your purines are going to be ag, A and G, adenine. They're purines because they have a double ring of carbon and nitrogen. Now, your pyrimidines are going to be cut, cytosine, uracil, and thymine. And they're pyrimidines because they have a single ring of carbon and nitrogen. Okay, so what's going to happen is you're going to have one purine and one pyrimidine making up each base pair. So A, for example, will bond with T. So you have your purine A bonding with your pyrimidine T. C will bond with G, your purine G bonded to your pyrimidine C. And then in uracil, the U comes in for RNA, and that will go where the T would be in DNA because you do not have T's in RNA, just like you do not have U's in DNA. So anyway, your two chains of nucleotides in DNA are held together by those hydrogen bonds between nitrogen bases. So, one base pair is the two nitrogen bases making up the same rung on the ladder. Now, you have someone called Shawgroff. His rule states that you will have equal amounts of A's and T's because they base pair. And the same thing is true for C's and G's. You'll have equal amounts in every DNA. Okay, so you might see a question asking you something along those lines. So like I said before, purines will always bond to or pair with a pyrimidine, and then your hydrogen bonds join the purine and pyrimidine. So like I said earlier, you might notice the structure of the purine is a little bit larger than the structure of the pyrimidines. And so you'll notice when we look at those pictures of the base pairs that you'll have one bonded to the other, okay? All right, that brings us to just one more go. C always bonds with G, so cytosine always bonds with guanine, and thymine always bonds with adenine, and that's in DNA. So if you take a look at this picture, this is just showing base pairing. So if you look here, for example, notice how the T and the A have a certain shape, kind of like the enzyme and substrate lock and key model, and the C and the G have a certain shape. Okay, how they could only bond to the other one. So again, with the base pairing rules, it's very strict. Your two strands are complementary. So if you know the sequence on one side, then you know the sequence on the other side. Because where there is a G, a C will always bond to it. Okay, so you're going to do a lot with base pairing for this unit. Be sure you know this for your test. Okay, so in DNA, if I ask for the other complementary side of DNA, this is how it will go. If I have a T, then an A will bond to it. If I have a C, then a G will bond to it. If I have a G, a C will bond to it. A will pair with T, A will pair with T will pair with A, T will pair with A. Okay, it's very predictable. So all organisms have chromosomes made of DNA of those nucleotides with A, T, C's, and G's. And this is how you are different because you put those nucleotides in a different order. So the sequence of nucleotides is what forms your genetic differences and all your genetic information for each organism. Okay, so this is what makes you different from a tree, for example. Okay, let's do some base pairing practice real quick. Okay, so one thing I forgot to tell y'all earlier is an easy way to remember it is apple in the tree, car in the garage. Okay, so A goes with T and C goes with G. Anyway, so we're just going to go through and do a quick DNA complementary strand. So this is DNA. Okay, so I'm just going to pull some letters out. That's supposed to be a G, sorry. Okay, notice I put them in groups of 3. You're going to see that a lot, and we'll talk about why later on. Anyway, so if I have DNA, and I know this is DNA because it has T's, and I ask you for the complementary strand, I just want the other side. So I know if there's an A, that a T will always bond with it. A T will always bond with A. A G will always bond with C. A C will bond with G, same thing. C, T, A, T. Okay, so what I just did is I made the second side of DNA. So again, here's a picture of DNA. Okay, so one more time, how you are different, how organisms are different is because they have a different order of nucleotides. Okay, so that order of the letters is what's different. Okay, so just a few things to go over. One chromosome, one of your 46 chromosomes might have 1,000 or more genes. One gene might have thousands of those nitrogen-based pairs. So as you can imagine, you get into a large number of nitrogen-based pairs. So the nucleotide sequence is that sequence of nitrogen bases, okay, and that determines what kind of protein is made. And the type of protein that's made then causes you to express your genes, to determine which gene is expressed, okay. So this is why every organism is unique, because their DNA is unique. And this is also why DNA is called the universal genetic code. Okay, every living thing has DNA, and we can look at that DNA and see the differences in it because of that. So again, your nucleotide sequence might be like A, G, T, C, C, T, A, G. That would be like a nucleotide sequence. That is what makes each of us different, because it might be a couple letters off. So that brings us to DNA replication. So first off, if you remember, we talked about when DNA replicates, and we briefly talked about what it was. So if you recall, DNA replication is simply taking one piece of DNA and making 2, or taking your 46 chromosomes and doubling that number to 92. So you're making a perfect replica or perfect copy of DNA. So we mentioned that it happens before mitosis and before meiosis in the S phase of interphase. So that is still something that you need to know. So that is going to precede or come before cell division, which is mitosis and meiosis. Now, you may also remember that prokaryotic cells do not go through mitosis or meiosis because they have no nucleus or no membrane-bound organelles. Their replication or their reproduction is much simpler. So when their DNA replicates, it's a little bit simpler. So if you recall, the only type of prokaryotic cell there is bacteria, okay? And they have no nucleus, no membrane-bound organelles. And so they have a circular loop of DNA, okay? And that's called a plasmid. So that's the circular DNA found in bacteria. And before it can divide into two new cells, then it has to replicate the DNA. So what happens is that replication moves around the bacteria in one direction because it is a circle. It might start at the center and then move in two directions, but it's going to go in a circular type pattern because the DNA is circular. And one cool thing is that because bacteria are much simpler, it only takes them about 40 minutes to replicate their DNA. So anyway, here's a great picture of your replication of the bacteria. Okay, so again, if you look, you have your one cell. Let me change to a different color here. So you have your one cell over here becoming two cells, right? Okay, so in order to do that, we've got to replicate our DNA. So first off, you have your circle, your plasmid, and it's going to integrate into the rest of the DNA. So plasmid is actually separate. So plasmid has some genetic components, but the main component would be in the red part. And then so it's going to integrate, and then it's going to replicate, and then there you get two new cells. It's pretty simple. So anyway, in eukaryotic cells, that would be us, we're going to replicate our DNA, and it's going to take much longer because you have a lot more DNA than a bacteria would. So for eukaryotic DNA replication, it's going to usually start at multiple different places, and it's going to spread out with lots of different replications going on at the same time. So, again, this might take hours to complete the DNA replication. And again, that's going to precede mitosis and meiosis, okay? So again, that's during the S phase of interphase before mitosis and before meiosis. Before your cells can divide, you have to make enough DNA to go in both of the new cells or all of the new cells. Okay, so replication by definition is the process where DNA is duplicated, forming two identical copies from the original. So the DNA strands will be identical to each other when replication is finished. So the steps to replication. So the steps to replication start out with an enzyme. And if you recall, enzymes have different functions, but they are going to speed up the reaction. So first off, the enzyme DNA helicase breaks the hydrogen bonds between your nitrogen bases that hold the two strands together. So therefore, it basically unzips the DNA. Okay, I remember this by helicase breaks the hydrogen bonds. Okay, so H goes with H. Helicase breaks those hydrogen bonds. Okay, and if you recall, the hydrogen bonds are between the nitrogen bases. Okay, so it's gonna, you had a bond between the A and the T. There's the bond. And helicase is gonna come along and it's gonna unzip it. Okay, and unzip that DNA. Replication only proceeds in one direction, from the 5' end to the 3' end. Anyway, so that would be step 1. So, step 2. As the DNA unzips, you have random free nucleotides are going to bond to the appropriate base. Okay, so I've split this A and T up here, okay? So now a free T is going to come and bond to this A, or free A is going to come and bond to that T, okay? And you can see eventually this is going to give us 2 DNA molecules. Then you have DNA polymerase, which is another enzyme. DNA polymerase comes along and makes these sugar phosphate bonds, okay? So again, P goes with P. Okay, so polymerase makes the phosphate and sugar bonds, connecting the rails on the ladder. So that pretty much finishes up replication. There are some other enzymes involved. It is more complicated than that, but that is all we're going to go over in this class. So anyway, yes, you can have errors once for every billion nucleotides because polymerase is error prone. But hopefully, when you go through, remember we talked about the checkpoints that you would go through. So hopefully when you go through the G2 checkpoint, which is after the S phase, after DNA is replicated, it will check it and either correct the problem or, that cell might go through apoptosis and die. So replication of DNA it doesn't start at one end and end at the other. It can happen at multiple different points along the way, which makes it faster, although it still takes a while. So now I'm going to show you a very simple drawing of how replication is going to occur. Okay, so I'm going to draw a really wide DNA molecule, because you'll see why in a minute. So here's my sugar phosphate backbone, and I'm just going to draw it kind of small. So we'll make this our A's, and then we'll say this is our T's, and then we'll put some C's and some G's. I'm labeling so you can see what they are. And then we'll put some hydrogen bonds in here. Okay, so we have our black, which is your sugar phosphate backbone, and then your gray, which is your hydrogen bonds connecting your nitrogen bases, and then your different colors of nitrogen bases. So, what's going to happen with replication is, first off, DNA helicase, the enzyme helicase, is going to come along and it's going to break all these hydrogen bonds. Okay, it's going to break all of them. I'm going to make us some space here to do what's next. So helicase broke the hydrogen bonds. Okay, so that was step 1. Step 2, free nucleotides are going to come up and bond. Okay, so what's going to happen is everywhere you see a T, an A will bond to it. Okay, so a random free A is going to bond to that T, and that free A is going to have a sugar and a phosphate attached. A sugar and a phosphate attached. Everywhere there's a free A free T will come up. And guess what's attached to that T? You guessed it, a sugar phosphate. I didn't make that one quite long enough. Let me fix that. I'm not an artist, as you can see. Everywhere you see a C, a G will come up and bond. And so all these free nucleotides are going to just continue filling in with their nitrogen base and their sugar and phosphate attached to it. Because remember, each nucleotide has one nitrogen base, one sugar, and one phosphate. Okay, so this is all going to happen simultaneously. Okay, but if you notice, our new strand is not connected yet. So that brings us to step 3, where polymerase is going to come along and connect your sugar and phosphate backbone. Okay, so where we started with one strand, now we have 2. So this side would be our old template. Okay, and then so we have our template, and then this side, both of these would be our new complementary strand. So here you have two different pictures showing replication. And first off, you notice that they're talking about a replication fork here. Okay, so that's where it forks right here. So anyway, you have both helicase and polymerase working here. And if you notice the leading strand, it's going from the 5' to the 3' end. Anyway, so you have multiple enzymes working. You'll see your polymerase here. We have another enzyme down here, ligase. Let me move it, but you don't have to know what it does. Anyway, so the hydrogen bonds were broken. It unzips. And then those free nucleotides are going to come up and bond. And so here you have this red strand, okay, which is going to be your new or complementary strand. And then your blue strand would be your original template strand. Okay, so again, here your new strand is going to be your complementary strand. You might also hear it called complementary, and the old strand will also be known as the template. Okay, and that's kind of what this slide is talking about. So replication is finished, complete when the entire molecule has been unzipped and replicated. So each new DNA will have one half of it that's from the original, the template, and the other half is new. It's the complementary strand from all those free nucleotides that joined up.